Hi everyone, in this video I'll be going over Lab 6, Gauging Confluency and Growth Kinetics. In this lab, you will be assessing the confluency of cells that you seeded previously and getting more practice passaging cells. Please also take some time to read the posted reading, as it may seem like a lot, but there's a lot of good information. So just please take a look. To kind of clarify, what we really want for you guys is not to kind of memorize all the really nitty-gritty details in these readings, but to kind of understand the big broader concepts and to be able to recognize words like confluency or contamination, what mycoplasma is, what mold is, and so forth. Okay? So anyways, for today's lab, we're going to be focusing on that concept of confluency. So simply put, confluency is the ratio between the surface area covered by your cells over the total surface area available. As detailed in these images, you'll see a percentage, the confluency, followed by the cell density in that image. And one of the consequences of confluency is that in these two extremes, so A and F, you'll notice that what's actually happening is in F, the cells are actually physically touching each other. And when this happens, something called contact inhibition occurs, and this can negatively affect proliferation of cells. On the other hand, in A, when there are not enough cells around, you don't have the proper growth factor signaling that comes from secretion of growth factors from one cell to the next. So you can kind of imagine that these two guys are talking to each other, but because this guy is so far away from everyone, he's not actually going to get the correct growth factor signals and he's going to die off. So it's important to kind of be in the middle of these two. So kind of ideally you want to be where C is. And whenever you're passaging, you never want your cells to get beyond 90% confluency or so, because at that point their behavior is going to be a bit different. Okay, so the procedure today is actually really simple. You're gonna remove your 6-well plate, you're gonna take a few pictures. You want to take a representative photo from each well, and this is more for your sake so that you kind of have a physical picture to go back to and be like, okay, so that's what seeding at 5,000 cells per centimeter squared and waiting 2 days look like. In this next part of the lab, you're actually going to passage them and you're going to count them, and then that way you'll be able to say like, okay, so this particular density, say 40,000 cells per centimeter squared, this is what a picture of that looks like. And really what we're trying to do is to help you develop your intuition for how you recognize cells and help you be able to see, if I seed so many cells at day 1, at day 5, how many cells will I have? And so you'll find that this particular skill is very useful for planning bigger experiments. Anyways, as far as actual tips and things go, please refer to the previous videos as well as the posted ones online. And I think that's everything for today as far as this lab procedure goes. I kind of want to spend a little bit of time going over the context of the next few labs. So in these first 2 cell culture labs, what you've learned to do is how to take care of your cells and how to seed them into different well plates and how to count them and so forth. What you're going to be doing next is learning different ways to figure out, well, how many cells do I have in these particular wells without passaging them and counting them under the hemocytometer. So those methods will include different chemical methods such as MTS, BCA, and a live dead assay. And so these are different methods for you to kind of assess the viability of your cells without having to necessarily perform something called an endpoint study where you actually kill your cells. So just kind of keep that in the back of your mind. And eventually what this is going to lead to is, for your independent projects, you'll be using, or you can use, I should say, some of these assays to determine if the experimental parameters you adjusted in your independent project actually affected your cell's proliferation without having to have a time point for each time. All right, so hopefully That makes sense. That's all for today. I'll see you guys next time.